Welcome to Chapter 4, the three-dimensional structure of proteins. In this lecture, we will be covering part 1 of this chapter. This part involves studying the properties of peptide bonds. The website shown in this slide is very useful when it comes to drawing a peptide. I strongly recommend you to go through this and draw some peptides of your choice. These are the learning goals for this chapter. First, we'll learn about structure and properties of the peptide bond. Next, we'll look at structural hierarchy in proteins. We'll also look at structure and functions of fibrous proteins. And we'll analyze the structure of globular proteins. Finally, we'll finish off with how proteins fold and unfold in solution. Let us revisit this figure from Chapter 3. The structure of proteins, as shown here, can be described at several levels of complexity, arranged in a kind of conceptual hierarchy. Right? You have the primary structure, then secondary, tertiary, and then quaternary structure. Primary structure is essentially a description of all covalent bonds, mainly peptide bonds that are formed between individual amino acids that are next to each other, and also disulfide bonds between cysteines in a polypeptide chain. The most important element of this primary structure is the sequence of amino acids that are arranged in a specific order. Secondary structure refers to a particular stable arrangement of amino acid residues giving rise to recurring structural patterns. For example, alpha helix or beta sheets, turns, etc. can be called as secondary structural elements. Tertiary structure, on the other hand, describes all aspects of the three-dimensional folding of a polypeptide. A primary structure goes to secondary structure. Secondary structural elements combine together to form tertiary structures. When a protein has two or more polypeptide units, so another polypeptide unit like this, right, when it binds to it, this arrangement in space is referred to as quaternary structure. So these are the assembled subunits and forms the quaternary structure. In this chapter, what we are going to look at is how these secondary structure elements are formed and how do they give rise to tertiary structure. One of the most important questions in the field of proteins is protein folding. The question is, how do a set of sequences, amino acid sequences, fold into a specific tertiary structural conformation? We'll see all these things in this chapter. Let us look at some of the key points that dictate protein structure. Proteins need to be arranged or folded into a specific conformation for function. What do I mean when I say folded into a specific conformation? The possible conformations of a protein or a protein segment include any structural state it can achieve without breaking covalent bonds. How can it do that? A change in its conformation could occur, for example, by a rotation about single bonds. Of the many conformations that are theoretically possible in a protein containing hundreds of single bonds, one or a few conformations generally predominate under biological conditions. And that is what I meant by saying into a specific conformation. And this is the thermodynamically and energetically stable conformation that a protein can adopt. And these folded conformations are often determined by the environment around the protein. When a protein adapts its normal function or structure, it is said to be in its native conformation. For example, a protein of your choice, when it functions the way it is expected to function, it is said to be in a native conformation. If it deviates from its function, it means that there is something wrong with its structure. So structure and function of a protein is closely related. The next question is, what drives protein folding? What are the factors that are involved in folding a polypeptide into a protein? Most native proteins are only marginally stable. Covalent and weak interactions drive the energetics of folding. We have seen what covalent bonds are, and we have also seen as to what weak interactions are, right? The van der Waals interaction, hydrogen bond interactions, ionic interactions, hydrophobic interactions, etc. Now, it is a difference in energy between the folded and unfolded states that help a protein fold, not the absolute energies. Like I said, a specific native conformation of a protein is the most energetically favorable conformation. Now, the energy, when I say energy, what I mean is the difference in the energy between the native, which is the folded conformation, and the unfolded state. Let us look at these forces that are involved in folding covalent bonds. Pretty strong interactions, right? Typically of the order of 400 kilojoules per moles. Weak interactions range from 2 to 50 kilojoules per moles. They are less stronger than covalent bonds. Now, you all know the equation for Gibbs free energy change, right? And it is driven by a change in enthalpy and change in entropy. Now, if you were to look at forces in folding, stronger interactions, which are the covalent bonds, include peptide bonds. Peptide bonds are the bonds between two amino acids. The CONH bond is a peptide bond. Disulfide bonds, shown here is a disulfide bond, right? Now, this, if you consider this as a polypeptide, this is a cysteine residue. The cysteine side chain dimerizes or reacts with a cysteine side chain of another polypeptide to form a disulfide bond. Essentially, the reaction that is happening is an oxidation reaction. When you break the disulfide bond, you're adding hydrogen. So it's a reduction reaction. Cysteines are reduced, whereas disulfide bonds are oxidized. Remember these. In addition, there could be prosthetic groups. Prosthetic groups are nothing but molecules that are attached to the proteins that are not amino acids. Non-protein, non-peptide molecules may be covalently linked, organic or inorganic. There are certain vitamins that are covalently linked to proteins. Sugars, lipids, and even metals, transition metals like iron, zinc, cobalt, copper are attached to proteins. Some of the weak forces include hydrophobic interactions, which is one of the most important interaction that leads to protein folding. Hydrogen bonds to water, internal hydrogen bonds, van der Waals interaction, electrostatic interactions. There are two different kinds of interactions, electrostatic interactions. One is a salt bridge, particularly those buried in hydrophobic environment, and interaction between permanently charged groups. These are the forces, right? There are two differences. One of them is the hydrogen bond that the protein forms with water, and also the hydrogen bonds that the residues in a protein form among themselves. Two different things. Van der Waals interaction is, as usual, any non-covalent interaction is dictated by van der Waals, right? Electrostatic interactions, you see the differences here, right? Salt bridges that are particularly buried in hydrophobic environments. and also the interaction between permanently charged groups. The peptide bond. The peptide bond is a very interesting bond and one that you definitely need to understand. This bond is formed between the carboxyl group of one amino acid and the amine of the next. Essentially, it forms an amide. The peptide or protein backbone refers to the C alpha, C CO, and nitrogen atoms of the amino acids and hence protein. The 6 atoms involved in backbone are C alpha, C CO, N, OCO, and HNH. These 6 atoms are coplanar. I'll explain it to you in the next slide as to what these 6 atoms are. But essentially, the backbone of a protein is the peptide bond. Shown here is a peptide in 3-dimensional space. Now, let me give you a perspective here. We'll start from the N terminus and go to the C terminus because that is how you read a peptide, right? From N to C. Now, as we go along the bonds, right? C alpha, C N, C alpha, C O, N H, C alpha, C O, N H, you see that there is a plane of the paper. The CN or the CONH bond is along the paper, which, or I can say it's along your computer screen. Whereas the C alpha R is pointing towards you, or it's pointing up. And the C alpha H, which is in the back side, as you can see, is pointing down, or it's away from you. This is the 3-dimensional perspective. Now, the 6 atoms that I told you about that makes the backbone, what are those atoms? They are C alpha, C, O, N, H, C alpha. These are the 6 atoms that form the backbone of a protein. The peptide bond or the amide bond has a property, and that is that the CON bond length in the peptide bond is shorter than expected. It is 1.3 angstroms versus 1.45 angstroms. Why is that? Think about it. This is a result of the amide having a partial positive and the carbonyl O being partial negative. This is one of the reasons why that CON bond, right, that is partially single and not completely a single bond. As you know, single bond is the longest bond. Double bond is shorter, triple bond is even shorter. But this is in between a single bond and a double bond. Let's see what it is. So as I said, the NH has a partial positive and CO has a partial negative, that is towards the oxygen. This is because of the resonance that exists in a peptide bond. As you can see, there is a lone pair on this nitrogen that can go to this bond, and then the oxygen can attract the double bond from the carbonyl towards it, and hence this bond is polarized, right? The electron pair is attracted towards the oxygen, and hence this is polarized and is partial double bond. It has a partial double bond character. Now, this kind of representation do not exist. What we usually represent is like this. When we write like this, but it is understood that this bond is partial double bond, and it always occur in trans configuration, virtually all peptide bonds. There are exceptions. Let us revisit this peptide structure again. There's one more thing that I would like to point out, that not all of these bonds can be rotated, right? Because this is partial double bond, so you will not be able to rotate that bond. What other bonds can be rotated? The NC alpha bond can be rotated, and it is dictated by the angle, the dihedral angle phi. It's the Greek letter, and that angle is between H, N, C alpha, and R. In addition, the other bond, C alpha C, in the same amino acid residue of the peptide, can be rotated, and it is dictated by the dihedral angle psi, and that is given by either R C alpha C CO or H C alpha C CO. Right, this is the dihedral angle. So, revisiting peptide bond rotations. C, this is the carbonyl C-N bond, does not allow rotation because it has a partial double bond character due to resonance. The C alpha N and the C CO C alpha bonds can freely rotate. Phi is the angle around this alpha carbon amide nitrogen bond, or C alpha N, and psi around the alpha carbon carbonyl carbon bond, or the C alpha CO. In a fully extended polypeptide, both psi and phi are 180 degrees, which usually is not useful for us, right? Because proteins are folded, and you do not want an extended polypeptide conformation. This slide gives you a perspective as to how psi and phi values change when the bonds rotate. Psi is between C alpha CO, and phi is between N and C alpha, right? Now, if you look at this rotation in this direction, viewer can see that when NC alpha bond rotates, as it rotates clockwise, the value of phi increases. The same with psi. If you were to look at the CO bond rotation. Now, as you rotate clockwise, it goes from 0 to 180 and then back to 0. As I said, 180 is the maximum that you can rotate, and when phi and psi are 180, the polypeptide is in its most elongated conformation. This leads us to the problem with bond rotation. As we saw in the previous slide, phi and psi can have any value between minus 180 degrees and plus 180 degrees, but many values are prohibited by steric interference between atoms in the polypeptide backbone and amino acid side chains. So in large proteins, amino acids can only adopt a limited number of phi-psi combinations. Some phi and psi combinations are very unfavorable because of steric crowding of backbone atoms with other atoms in the backbone or side chains. Some phi and psi combinations are more favorable because of chance to form favorable hydrogen bonding interactions along the backbone. So, the conformation in which the phi and psi are prohibited is because they have steric clashes. That's the bottom line that you get from this slide, right? And those conformations that are favorable are because they can form hydrogen bonding internally and stabilize the conformation. So the next question would be, what are the allowed values of phi and psi angles for a peptide or a polypeptide or a protein? The allowed phi-psi combinations for an amino acid or a peptide can be graphed on a Ramachandran plot. Now this is a plot that shows psi on the y-axis versus phi on the x-axis. And as you can see, the plot goes from minus 180 to plus 180 on the x and minus 180 to plus 180 on the y-axis. Right? Now this was first introduced by the scientist G. N. Ramachandran. and this was deemed very useful for predicting the three-dimensional protein structures and the secondary elements in the three-dimensional protein structures that are allowed and disallowed. This slide gives you an example as to how you can look at the Ramachandran plot for a specific amino acid residue and predict as to which conformations are allowed and not allowed. Now, this is an alanine residue in a polypeptide. The polypeptide continues here and continues here. Right? This specific conformation where the R side chain is up, this is a methyl group because it's an alanine, right? It's up, and the carbonyl is down and NH is up. This is an allowed conformation that is given by a specific psi value and a specific phi value, and it would be 120 for psi and around minus 120 for phi. This conformation where this CONH is flipped, right? The CO goes up or goes behind the plane and NH goes up or coming towards you. This is also an allowed conformation. However, the psi and the phi angles change. And in this case, the psi is minus 60 and the phi is close to minus 50, right? Whereas this kind of a conformation where phi and psi are both 90 and minus 90 degrees respectively is disfavored because when you rotate the molecule, the CO would sterically clash with the methyl group, and that is disfavored. So this is how Ramachandran plot helps you in predicting the feasibility of a specific psi and phi value for an amino acid residue in a peptide chain. Before I end this lecture, as a final slide, I would like to introduce you to the secondary structural elements of a protein. Secondary structure is a local organization of the primary structure, which is nothing but the sequence of a peptide or a protein. Secondary structure is limited by the phi, psi angles, side chain composition, and all the weak interactive forces. Major classes of secondary structural elements include alpha helices, beta sheets, beta turns, and random coil. Random coil is a structure where there is no specific orientation of the side chains of a peptide, hence the name random coil.